Hi everybody, in our last video we started to talk about the structure of muscles and muscle tissue. Very quick recap. Okay, we said we have repeating alternating units, okay, as you can see in this longitudinal image of striated muscle. Okay, alternating from light into dark, back into light again. Okay, remember one sarcomere runs from one Z line through to the next Z line. So this section here is classed as one sarcomere. And that sarcomere is made up of a thin actin protein and thick myosin protein. So basically contraction and relaxation of the striated muscle is controlled by the action of these two proteins and how they interact with each other. Okay, we're going to look at that now. Okay, now as a skill, it's as part of the new syllabus, students are required to draw the sarcomere, okay, and this is basically how you should draw it. Try and obviously simplify it a little bit, but there are some really key things to remember. So, just to get it straight again, all right, and allow it to sort of look from this to the other diagram and kind of make the comparisons between them. We have a Z line, okay, now along that Z line, that's here, all right. All right, here's one other Z line. This is here. Now what you can see, this Z line is also made up of actin filaments. Okay, so it's very important to remember. You've got a Z line of actin filaments here. You've got the Z line, you've got these parallel lines of thin actin running parallel or alongside. Okay, these remember quite thin, and in the middle you've got the thicker myosin filament proteins, okay? And it's important to remember that for every one myosin, so every one of these, okay, next to it you've got 1, 2, 3, 4, 5, 6 actin filaments, okay? And what we can say is that the myosin fibers are interdigitated with the actin, okay, and make up that dark band. Okay, so what we mean by interdigitated is that it's like, imagine taking both your hands, interlocking your fingers, okay? You're interlocking your digits, and that's what this interdigitated means. I've never heard of this word before. I had to search it up, just read it in the book, okay? But I thought it was quite a good way to kind of explain what is happening, okay? So we've got the light band, which makes up our Z line and actin, okay, here. And then we've got our myosin, which is interdigitated with actin here, which makes up the dark band. And the distance between one Z line and the next Z line, remember, we call it our sarcomere. Okay, so when you're drawing this, you've got to remember that a sarcomere is between the two Z lines. You should always draw your myosin filaments with these little knobbly heads, because they're really important. So we're going to come back to them, okay? These little heads that we can see, okay, allow the myosin to sort of grab hold of our actin filaments, okay, and move along. So that's really important that you draw them with these little knobbly heads on them. The actin filaments that we can see, these ones here, should be connected to the Z lines, and light bands should be labeled around the Z line. So this should be always labeled as the Z line, as the light band. And the extent of the dark band, obviously, which is where the two proteins here overlap, should be also indicated on your diagram. Remember, guys, as it's new to the syllabus, it's something that they're likely to put into the exam. Okay, so learn how to do this, practice the skill, make sure you remember where all the different parts go. All right. Okay, so let's have a look at the mechanism then by which muscles contract. The first one, probably the easiest diagram to explain here is seen in this one. So what happens during contraction is that the myosin filaments, so these are the myosin filaments with their knobbly heads, okay, they're going to pull against the actin filaments. So here's your actin filaments here, here's your Z line, which remember your Z line is also made of actin, all right? So this is 2 fibers of actin, 2 fibers of actin, 2 fibers of actin. Your myosin is going to grab hold, okay, on each end of these fibers, okay, and it's going to pull them, and it's going to cause a sliding. So when it's relaxed, you can see this, the light band is quite long, okay. At the moment, this is not contracted, And so the light band is much longer, okay? And the dark band, okay, is here in the middle. Now look, when it's contracting, look, the myosin heads are pulling against the actin filaments on both ends and pulling it in, okay? And so what's happening here is that they're getting the light bands, so the band that's just made up of actin is going to get smaller, okay? And there's going to be more space that's overlapping with actin and myosin here, okay? So this light band is going to shorten, okay, indicating that the actin is sliding along the myosin, and the dark band, okay, is going to sort of remain the same because the dark band is only as long as the myosin molecule itself, okay? So that's going to remain the same length wherever we look at it. Now over here, this is sort of showing an idea of how this is happening. Okay, so here's two of our proteins. Okay, this bobbly-knobbly one over here is actin. This grey one is myosin. Okay, and these little bits here are the myosin heads that you can see. And so what these myosin heads do is they connect to a binding site on the actin, okay, causing formation of what we call a cross bridge. Okay. But this only happens in the presence of calcium ions. And again, we're going to come back to that idea of calcium ions and when these are released. Okay, so this is only happening when the sarcoplasmic reticulum is kind of stimulated by a sensory neuron, by a motor neuron, okay, causing a release of the calcium ions, which allows the myosin head to bind to the actin, okay? So we're going to come back to that again. I'm going to keep coming back to that idea because it's really key to understanding what is actually happening here. All right. So formation of this cross bridge, and then you've got the movement of the actin as it slides along, okay, connected. Okay, this will then be released, and this will then join into another binding site further up, okay, which is what we're going to look at in a second. So our next question is, first of all, let's have a very quick look at this. Again, you've got to be able to do a relative relevant skill that you've got to remember about and how to do is figure out whether a muscle fiber is a contraction or relaxant. Okay, now remember, when it's a muscle fiber is relaxed, it means the sarcomere is going to be longer. Okay, the white area, okay, or the light band is going to be much longer than when the muscle is contracted. So now you can see here in the contracted sarcomere that these areas are narrower, okay, the dark areas stay the same. So if you've got two images and you've got to compare the two, which you'll probably have to do, okay, you can much more easily make that comparison, all right? So just a very quick thing to be able to do. Okay, so I'm going to just come back to this one again because we're going to talk very quickly about muscle contraction and the role that calcium ions kind of play in that. So you can't really see it very well on these images, but in relaxed muscle, you have a regulatory protein called tropomyosin that blocks the binding site on the actin. So you imagine this, when it's relaxed, there is a protein here which blocks this binding site. And that's called tropomyosin. Now remember, what's going to cause the muscle to contract is when a signal from a motor neuron, so an action potential from a motor neuron, reaches the muscle. Now if that happens, the sarcoplasmic reticulum, remember, which is that kind of evolved endoplasmic reticulum, is going to release all that calcium that it stores up. Okay? And what these calcium ions do, they bind to another protein, okay, called troponin. And that troponin causes the tropomyosin, which is the blocking protein here, to move away, exposing the binding site, okay, which is here. But only when that binding site is free, okay, will the myosin head be able to bind and swivel and join to it and form that cross bridge, okay? So that's when the two join together. Now, as you can see here, what's happening is this is going to join to here, and it's going to swivel along, and it's going to be pushing the actin in this direction here, okay, causing it to kind of get smaller as it moves further towards the sort of the center of the dark band, okay, and that's what we can see happening here. Okay, there is going to be one more short video on the role of ATP in the sliding filament theory. Thank you.